everybody, my name is Torres and I'm here with the Honors College at Star Kingwood campus. And today I want to talk about my experience with tiny earth research in my biology classroom. So did you guys know that beneath this surface lies secrets to survival in the soil? And these remarkable microbes produce antibiotics that exhibit antibiosis that compete against other types of bacteria. And so I want to talk about the microbial showdown of how two antibiotic producing bacteria can compete with each other in vitro testing. So because of this interaction, it led me to my question of how would two different antibiotic producing bacteria respond to one another in the same soil sample. So as I referred to earlier, antibiotic producing bacteria does form antibiosis, and you guys might be wondering what antibiosis is. So antibiosis is the biological interaction where one organism produces substances such as toxins, antibiotics, and other chemicals that inhibit the growth of other microbial organisms. Okay, so for the experiment, we decided to get soil samples from different areas. So I used one of my soil samples right here that I got from a nearby lake, and then I used two other soil samples from my peers in my biology classroom, one from a backyard and another one from a creek. So before I get into my whole experiment and just start throwing out random words, I want to explain to you guys the materials that I used for this experiment. So by collecting the soil samples, I did use a microcentrifuge tube to put the soil samples in, and we refrigerated them, and then we added saline to make a type of slurry mixture, and then from there we used a micropipette to take some of the slurry, and then we put it into one of the 4 microcentrifuge tubes to create dilutions. So we put 900 microliters of deionized water in 3 of the microcentrifuge tubes, and then just purely 100 microliters of this slurry of just the soil and the saline in the first microcentrifuge tube. And then we would use the micropipette to take from the first microcentrifuge tube to the second to create dilution, and into the 4th one, diluting it further. And then I will also be using an inoculating needle and inoculum loop right here for the transfer of bacteria from my plates to make patch plates, streak plates, and for the other experiments. And then finally, I will use a cork borer for one of my experiments that I will be showing on a little later. And I want you to pay attention to right here. It's hollow, so whenever you stamp it inside of the plate media, it will create a sort of stamp, and you can take out that circular media and then place it onto different medias. Okay, so as I said before, for my dilution plates, I did use the micropipette and the microcentrifuge tubes, and this was my first microcentrifuge tube dilution, which was the most innumerate one that contained between 30 to 100 microbial colonies. And then from this, I used a needle to pick out certain colonies that looked very different characteristically, and then plated them on a patch plate right here, 16 of them to be exact, so I can isolate these microbial colonies. And to isolate them, we want to make sure that it's a pure colony and not just a mixed culture. And then from this patch plate, I tested against 3 of the escape safe relatives, B. subtilis, S. epidermidis, and E. coli. And then, as you can see right here, there seems to be signs of antibiosis. It is creating a sort of barrier around that isolate as well as one, and you can't really tell in 16, but it is as well. So I decided to take 2 of my isolates that were showing a response to antibiosis, and so I picked my isolate number 3 and then isolate number 16 and made a streak plate with using the loop so I can make sure that it is for sure not a mixed culture. And then after this, I used the cork borer that I explained earlier that had the hollow circle at the bottom to make plugs from my different isolates, as you can see here, and then put them in 3 different plates right across from each other. But I also had to have a negative control with LBA plugs put right here, and then positive control for my L-antibioticus plugs right here. However, with this experiment, I failed to put a good proximity between the 2 isolates, so it didn't really show any type of interaction since they were too far apart. So that led me to go to a different method, which was the streak method. And right here, I first put isolate number 3 vertically across the plate, isolate number 16 vertically across this plate, incubated that, and then after it grew, we did B. subtilis as the negative control right above, and then positive control L-antibioticus, and then the other isolate that I'm testing against right down the middle. So as you can see right here, it is showing proof of antibiosis. As you can see, the barrier that's forming against the B. subtilis from the isolate number 3, but you don't see that happening with the 2 antibiotic producing isolates, and you can see that it's just going right through each other. But since it is not inhibiting any type of growth, it is a type of interaction that they are showing. And then unfortunately, with my positive control, L-antibioticus, it doesn't seem to be any growth, and that could be either because I didn't put enough of the bacteria on my loop, or it didn't have enough time in the incubator for it to grow. And so with this, I concluded that there is no signs of antibiosis between 2 antibiotic producing bacteria in these soil samples. However, because of human error, I would like to do further research on this and do different methods. And then I would actually like to look into the environmental factors such as pH levels, temperature levels, and nutrient availability. And so with this, I want to see if there's a different type of pH or a different type of temperature that it will kind of determine if different types of antibiotic producing bacteria grow within the soil. And then I would also like to look at different antibiotic producing bacteria and how they respond to either the same organisms or different organisms. And that's why I'm excited. And thank you.